This lecture is on photosynthesis, and it's found in chapter 10 of the Campbell textbook. So, first of all, life comes from light and air. That's the statement we're going to begin with. And essentially, all life on Earth is powered by the sun, so it's solar powered. To begin, we should look at a review of the two basic types of organisms in an ecosystem, in all of life, in fact, in the biosphere. And all of these organisms, no matter which group they fall into, need a constant input of energy in order to function and for the cellular processes to occur. So we have, first of all, the heterotrophs, which are like us. They're animals, they're the consumers, and they get their energy from the hetero part, eating others. Basically, that means they eat organic molecules that have been made by other organisms. And then they get their energy through the conversion of those organic molecules into energy through respiration. The other group, the ones that actually make life able to sustain itself and have the energy it needs, are autotrophs. And those are our plants and our producers. They produce their own energy from CO2 and inorganic materials that they obtain from the environment, and they convert the energy of sunlight to build or go through an anabolic process of building organic molecules. And this is done through primarily photosynthesis, although we do have a few other types of methods for that to occur. What we have here being made by the autotrophs are organic molecules, which in turn are the ones that are going to be used by heterotrophs to get their energy. So on a molecular basis, how heterotrophs and autotrophs are related comes down to the chemical formula that we are familiar with when we talk about photosynthesis or cellular respiration. So heterotrophs make their energy in organic molecules by eating organic molecules made by others. And those molecules get broken down into something usable like glucose, which when combined with oxygen in this formula produces the products of carbon dioxide and water, and the most important product, ATP. In this type of reaction, we have oxidation occurring. So the glucose molecule is oxidated to the CO2. The carbon is oxidated down into the CO2 there as a product. And this type of reaction is a catabolic reaction that is going to be exergonic. The autotrophs, on the other hand, are going to make their energy in organic molecules by converting light energy and using that light energy to reduce CO2 into an organic molecule like glucose. So that reduction, anabolic type of reaction, the building type of reaction, is endergonic. It requires energy, and that energy is, of course, going to be light energy in the case of photosynthetic organisms. So we're going to focus on photosynthetic organisms, those that do use light in order to convert their energy into organic molecules. And those are going to primarily be plants. So one of the structural parts of a plant that you should be familiar with is the stoma or stomata that allow the passage of CO2 and water in and out of a leaf. The leaf is where most of photosynthesis is going to occur. It's where the majority of chloroplasts, where photosynthesis actually takes place, are found. even though you can find chloroplasts in other parts of a plant. But the leaves are really going to be the site where most photosynthesis is going to occur. So in the mesophyll of a leaf that you can see there on the right, you're going to find the chloroplasts lined up. And there are probably between 30 and 40 chloroplasts in every single plant cell in a leaf. So you can see in this cross section of a leaf right here where the chloroplasts can be found in both the palisade layer and also in this spongy mesophyll here. So there are lots and lots of chloroplasts found in a leaf cell. And basically, photosynthetic enzymes and other molecules are grouped together in a biological membrane found within the leaf, which allows the necessary series of chemical reactions to be carried out efficiently. Because of this, there's a thought that the process of photosynthesis likely came about through a group of bacteria that had enfolded regions of a plasma membrane that contained these same clusters of molecules that are involved in the photosynthesis reactions. Today, part of the evidence that we use to make that assumption is that in existing photosynthetic bacteria, you can find enfolded photosynthetic membranes that function similar to a chloroplast and the internal membranes that you will find in a chloroplast. So because of this, we have the endosymbiont theory, which suggests that the original chloroplast was actually that type of photosynthetic prokaryote that eventually came to live within a eukaryotic cell. So our major organelle that's going to be involved in a eukaryotic plant cell in the process of photosynthesis is going to be our chloroplast. All green parts of a plant have chloroplasts, but leaves are the major sites of photosynthesis for most plants. I already mentioned that there are 30 to 40 chloroplasts in every plant cell, and there are about 500,000 chloroplasts in a square millimeter of a leaf surface. Each chloroplast has two membranes, so it's a double membraned organelle. Suspended within that membrane is a dense liquid that we call the stroma. And then within that stroma you'll find a system of sacs called the thylakoids. It's within these thylakoids that you actually are going to find chlorophyll, which is the green pigment that we see in leaves and in plants. The stacks of thylakoid are called grana, or if you have one, granum. And we'll look more carefully at the different reactions in the parts of photosynthesis that occur in these different structures. So here's another picture showing chloroplasts, and actually zooming in from the macro view of a leaf down through the mesophyll of that leaf's cross section there, and then into the actual plant cell where we're going to find our chloroplasts. And what I wanted to point out is, first of all, we're going to be looking at both the light-dependent reactions and the light-independent reactions in photosynthesis. And the first part, the light-dependent reactions, are going to be occurring in the thylakoid membrane. So if you look here at our thylakoids, you can see they do look like empty sacs. And within that membrane is where you're going to find things like the chlorophyll molecules. And this is where the electron transport chain is going to be doing its work. And also where ATP synthase can be found. So all of those things are occurring within the membrane. So you can see how with this membrane having all these folds and this sac-like structure allows it to have a lot of surface area. There's a real amplification or multiplication of surface area just by the structure of these thylakoid membranes and thylakoid sacs. One other thing I want to point out with this picture is that there is also this empty area within the thylakoid, which is going to play a part in some of the reactions. And we call that the thylakoid space or the thylakoid lumen. In between the two membranes of the chloroplast, there's another space that you should be aware of, and that's called the intermembrane space of the chloroplast. So those two sets of reactions, again, we have the light reactions and the light independent reactions. So the light reactions are what occur first. This is that harnessing of energy that is going to take the light energy and begin to store some of that energy in molecules that will later be used by the Calvin cycle, which is what makes up the light independent reactions. So the first set of reactions, the light reactions, are the ones that are going to be converting solar energy into chemical energy. So essentially, energy from the sunlight is used to split water, which provides a source of electrons and protons in order to then activate ATP and NADPH, which are electron carriers that take that electron energy over to the Calvin cycle. And the Calvin cycle, which is the light independent reactions, are the ones that are going to be building, or the anabolic reactions, building sugar. The Calvin cycle is named for Melvin Calvin, who with his colleagues worked out many of the steps in the 1940s. What happens is that the ATP and NADPH, the energy from those electron carriers, is used to reduce CO2, which is taken in through the stroma of the leaf, stoma actually, of the leaf, and that process of incorporating the gas, the CO2 gas, into something that is an organic molecule, solid molecule, is called the fixing of the gas into the solid, and that occurs in the Calvin cycle. So looking at our reaction for photosynthesis again, it's important to see where our products and reactants are entering and exiting the photosynthetic process in the chloroplast. So the first thing we have here is that we need CO2. So the CO2 is actually not going to be used until the Calvin cycle, so not until we get into the light independent reactions. And at that point it becomes reduced using the electrons that have been obtained through the light reactions. So even though we say that the Calvin cycle is light independent, actually most of that cycle and that process does occur during daylight hours because there's a steady need and requirement for the electrons that are being carried by the NADPH and the ATP in order to fix carbon from CO2 into this organic molecule, or the building block for the organic molecules of glucose. The other reactant that we need is